So bacterial normal flora, these are a variety of microorganisms. And for today, they are bacteria, the microorganisms, and they are harbored in some anatomic parts of humans. And at that time, for them to be normal flora, there's no disease manifestations resulting from their presence in those parts under normal circumstances. So there are two categories of organisms that are found on the body that fit into that description. One group is referred to as transient flora. And these ones inhabit the skin or mucous membranes for a limited duration. They are derived from the immediate environment. So it could just be lying somewhere, it was deposited, and then somebody comes by and it stays on that person, but for a limited duration. It can be days, it can be hours, it can even be weeks, but eventually they disappear. But it's important because they can cause disease when there is disturbance in the defense mechanisms in that part at that particular time when they are present in that part. Then the second category are the resident flora, and these are the ones we are going to say a little bit more about. These are relatively fixed types of microorganisms regularly found in specific anatomic parts. And if they are disrupted in one way or another by scrubbing or washing, they reduce, but they reestablish themselves after a short time. So they are fixed, kind of relatively, not tightly. So the first lot of this group is acquired by the newborn baby during passage through the birth canal. In the uterus, the baby doesn't have them. But when it comes out, it's going to pass. The ones that are born vaginally, they're going to pass through the birth canal of a mother that already has some. So it acquires some. But even the ones that are not born vaginally, they eventually get these organisms from those handling them, from the immediate environment. So they also start accumulating them. So subsequently, the organisms are acquired from other humans and inanimate sources in the environment. So the distribution of resident flora, there are a number of factors that influence their distribution, both in types, which type is to be found where and in what quantity. So there are some mechanisms that determine those, and then there are some mechanisms that regulate the quantity so that they don't exceed a certain limit. So the distribution is partly influenced by the general physiological characteristics, including the body temperature in various parts. So there are some parts which are warm, and there are some organisms that prefer those warm areas. And then there are some parts of the body which are cool, and there are some types of bacteria which are found in those cooler parts. Then the body also has secretions on the skin and mucous membranes, and depending on the content and the pH of that secretion, it will determine which organism is going to be found there. And then availability of oxygen. There are some that prefer large quantities of oxygen. There are others which cannot withstand large quantities, so they'll be found in hidden parts. And also moisture, availability of moisture. Some parts are drier than others. There are some that prefer areas with a lot of moisture, others that prefer some areas with little moisture. Then there are also other characteristics which are important, like age and sex. And these, especially the sex, we mean here gender, male or female. That is what we mean by sex there. And the female, there are some characteristics, biological characteristics which change with age. and they also affect or involve the hormonal activity. Even the hormones, they change and the activity change, and all those would add up to determine which organisms are going to be found in which particular female, in which particular part of the body. So there, although we said they are fixed, we said it's relative. So they are associated with minor fluctuations in quantity and types throughout life. For example, there are few in early childhood and they increase with age. So the parts, there are some parts which are free of this normal resident flora. And some people describe them as sterile, but they're not really sterile. They're just considered to be free of resident flora. And if you look at the respiratory system again, the lower respiratory tract, starting from the trachea, the bronchi, the bronchioles, and the alveoli. These have cilia or inbuilt mechanisms like secretions of mucus that would catch any bacterium that might land there, and eventually it gets expelled. So the lungs are totally free of normal flora and bronchioles and bronchi and up to the trachea. Then as you go upwards, you come to the upper respiratory tract, which we are going to mention in the other part. So this lower part of the respiratory tract, there are various defense mechanisms that clear microorganisms that may enter. I think I've already mentioned ciliated epithelium that moves mucus upwards, and phagocytosis of microorganisms by phagocytic cells. Then secondly, gastrointestinal tract. It's a long tract, but there are parts which are considered free of normal flora. First, the esophagus, that is the resident flora. Naturally, it's considered free of the resident flora, but it may contain bacteria from ingested food or from saliva being swallowed from the mouth, and those are encountered there as transient flora. Then the stomach, the pH there does not favor most bacteria. So the acidity plays a protective role by keeping the number of the organisms at a minimum. Then the upper part of the small intestine, this is in close proximity to the stomach, and the contents are more or less like what one has in the stomach. And then genitourinary tract, the upper part, that is the kidney, the ureters, and the bladder, they are free of normal flora. The bladder might get some transient flora by upward mobility, but naturally it's free of resident flora. Then in addition to those, there are also other parts that are worth mentioning when it comes to being free of normal flora. The blood circulatory system. It is free of normal flora. Again, this from time to time, one gets what is called transient bacteremia from the normal flora, but it's just transient flora again. But resident flora are absent in the blood. And then the central nervous system, the specimen that is encountered from there is cerebrospinal fluid, and there should be no bacteria in it as part of the normal flora. And also the peritoneal cavity, the peritoneal fluid should have no resident normal flora. So these are important because any organism isolated from a sample of properly collected and properly handled specimen from these sites, the blood, the CSF, the peritoneal fluid, should be significant, even if it is an organism that is usually regarded as normal flora elsewhere. And what is emphasized in addition to that is that the specimen should be collected as septically as possible, so that one does not introduce normal flora and then misinterpret the findings. It will cause a lot of confusion. So with that, we go to some parts which are regarded as are well known to harbor normal flora in large numbers. The skin. This is the external part exposed to the environment, and it is supposed to harbor quite a large number of organisms as resident flora. It also harbors transient flora because it's a part that gets into contact with the environment. And on the skin, there are many organisms that can be found, quite a variety. Gram-positive cocci are the dominating ones, and some gram-positive bacilli as well, but not as many as the gram-positive cocci. The types and distribution of these organisms on the skin are influenced by microenvironmental factors, some of which we had already mentioned, including availability of moisture, and then various regulatory mechanisms which keep the number and types in check. Among the regulatory mechanisms, you have some physiological mechanisms. There are some that make it unfavorable for other bacteria. There can be secretions with low pH, and sebaceous secretions also contain fatty acid and enzyme-like substance referred to as lysozyme. And this enzyme destroys bacteria, it is bacteriolytic. So on the skin, sweating and washing or scrubbing do not eliminate the normal flora. The number may be reduced temporarily, especially if it's vigorous scrubbing with soap or antiseptic, but the organisms reestablish themselves as soon as after that. So the skin normal flora are important because for a number of reasons. Among them, there's increased risk of contamination of specimens. These could be specimens collected from the skin itself or by going through the skin. And then there's also increased risk of introducing the normal flora into inner tissues through clinical invasive procedures or that specimen collection passage through the skin. And then they are also important because they pose a risk of infection or initiating a focus of infection associated with devices inserted through the skin and left in place for a long duration. And they are also encountered quite often as causative agents of wound infection. So proper skin cleansing is important before penetration. This would occur when one is giving an injection or collecting a specimen or for any other reason. So from the skin, we go to the nose. So the organisms which are found on the skin as normal flora can also be found in the nose. And one important organism is Staph aureus. And this is important because there are some people who harbor it. A certain percentage of the population harbors Staphylococcus aureus in the nasal cavity, and they can act as a source of infection, maybe to themselves or to other people. And then the mouth, again, it's an area which harbors quite a number of resident flora. Gram-positive cocci predominate there again, and the specific gram-positive cocci you will cover when you do the specific organisms. Then there are many others, lactobacilli, bacteroides, all those you will cover again when you are doing the specific organisms. But in addition, there is also a certain type of microorganism, which is not bacterium, which again you'll cover much later towards the end of the lectures. And that is the yeast Candida albicans, which is carried by some people in the mouth as part of the normal flora. Then the upper respiratory tract, also called pharynx, and when one moves closer to the mouth, it becomes oropharynx. It has the same organisms that are found in the mouth. And there are the genera, which you will encounter as you go on with the course. So it's also quite rich. It's just bordering the part we had talked about as being free of normal flora. But the mouth flora and the upper respiratory resident flora, they are potential causative agents of disease locally and in other parts. And the disease causation may or may not be due to alteration in immunological defense mechanisms. Large intestine, again, this is one part that is very rich in microbacteria of different types, many types, 100 different species. And they are basically bacilli and cocci. And these organisms are important because of the risk that they pose. That is risk of causing disease, one, in adjacent tissues as a result of disturbance, which could be in association with trauma or could result from trauma. Then they also become causative agents of disease if in one way or the other, or for one reason or another, they are relocated, they are removed from that area into other tissues. Then they no longer found there as normal flora, they begin to cause harm. So they become disease-causing organisms. So treatment or investigative procedures are performed applying the necessary precautions to minimize chances of relocating these organisms to other parts. And then another significance, it's quoted that there are some metabolic activities that these organisms carry out that may be beneficial to the host. Maybe they can destroy some substances which might be toxic, or they might stimulate immunological mechanisms, or they might synthesize some nutrients that are useful to the human body. Then the lower part of the urethra, generally it contains a small number of bacterial flora. We have talked about the upper part being free of normal flora, but the lower end, there are few organisms there as normal flora, and their significance is that sometimes they come out in the urine, a urine specimen that is being collected for examination. So they contaminate that. Then the lower female genital tract, the dominant flora is determined by the pH. And this pH also depends on hormonal effect, and the hormones depend on the age of that female. So the types of bacteria and dominant organisms are different. Soon after birth, in young adults, and in older females. So there's a lot of changes that go on depending on the age. But what is known for sure is that at any age, the dominant flora may be either lactobacilli or mixed cocci and bacilli. Then another important aspect of the flora in the lower female genital tract, the organisms from the large intestine. They are also encountered there as part of the vaginal flora. And this is due to close proximity with the opening to the large intestine. So the lactobacilli, which we mentioned, at that point when it is dominating, it maintains the acidic pH in that part. And that pH is unfavorable for would-be pathogens that would colonize the area and create an infection. This pH comes from their activity on glycogen. Is somebody talking in the background? No. So the significance of lower female genital tract flora, one, protection locally against common pathogens, and that is through the effect of the lactobacilli that we have just mentioned in creating a pH that is unfavorable for other organisms. Another significance is that we had mentioned a baby acquiring normal flora, those that are born vaginally, but they can also acquire organisms which are harmful to them. So the part of the normal flora can go into the baby and can create an infection or cause an infection in the early part of life because of what that baby acquired through the birth canal. And thirdly, the organisms from the intestinal tract, which we mentioned from time to time encountered there as part of the normal flora. They are significant as causative agents of urinary tract infections and infections of other pelvic tissues. So the opening to the female urinary tract, again, is close to that area, and it is also short and it's in contact. So one would get urinary tract infection, and most of the organisms isolated from that urine would be the organisms that are encountered in the large intestinal tract as normal flora. So in general, the significance of normal flora, there are some which are useful or beneficial. One is that they compete for microenvironments in various parts more effectively than some regular pathogens. And so they can occupy an area before the pathogen, so that the pathogen is not allowed, does not establish itself. So that is a way of providing protection by preventing colonization by the pathogens. Secondly, there is continuous stimulation of the immune system. They enable it to mature and to be active. And thirdly, there are some metabolic activities which may be useful in various ways. They could have protective effects, and they could also have byproducts which could be useful nutrients. And then there are harmful effects of normal flora. So normal flora in general can be potential causative agents of disease, and that they can cause disease depending on quite a number of circumstances. One, they can be opportunistic pathogens. And the examples of disease causation as opportunistic pathogens, some are seen in the mouth. We say there are very many organisms in the mouth. So some affect the teeth, some affect the mucosal surfaces and the gum, depending on the predisposing factor. Then they can be opportunistic infection of an opportunistic pathogen in damaged tissue. And there are also some physiological and other abnormalities in the humans which reduce the immunological defense mechanisms. And that would predispose that person to infection or disease that would originate or can come about because of the activity of the normal flora. So potential causative agents of disease as opportunistic pathogens, we have mentioned those three examples, but they can also be causative agents of disease as a result of relocation from the usual occupied site to another site. And thirdly, they can cause disease as a complication of prolonged administration of broad spectrum antibiotics. And that is this one example there that again you'll encounter as the course goes on. The antibiotics would clear the organisms that are susceptible to it, even the normal flora, but there are some that are resistant to it that would gain, would multiply because some of them have been eliminated and they would reach a proportion that would produce manifestations. And secondly, apart from being potential causative agents of disease under various circumstances, these organisms also contribute in the spread of antibiotic resistance. They are bacteria, just like any other bacteria, like in the gut. They live in close proximity and are capable of exchanging plasmids. And some of those plasmids code for what we had said earlier, resistance to antibiotics, and it can be shared among many. So when the bacteria originating from those ones infect a person, then there will be resistance to antibiotics. And I think that would be the last slide for today. And I say thank you very much, and also for enabling this arrangement to work. I had a lecture in the morning, which did not take off because of those microphone issues.